Hello everyone, I am Suvarna Gaikwad, Assistant Professor in रयत शिक्षण संस्थाज दादा पाटील महाविद्यालया कर्जत. So, welcome you all in the T.Y.B.Sc Paper-II Cell and Molecular Biology. So friends, today we are going to start new topic, that is gene regulation in prokaryotes. So let's start. So friends, as we know, gene expression is highly regulated process, and gene regulation is nothing but any mechanism used by a cell to increase or decrease a particular product's production. Okay? So this gene expression in prokaryotes is largely regulated at the point of transcription. Okay? And regulating this gene expression in prokaryotes ensures that a cell's resources should not be wasted by making proteins when that cell doesn't require that protein. Okay? Let's discuss the operon concept. So in 1961, Jacob and Monod, they proposed a mechanism called as operon model. This model is for regulation of gene action in E. coli. So the operon is a part of genetic material or part of DNA. Now the genomic DNA of prokaryotes contains both types of genes, that is structural genes and regulatory genes. Let's see the transcriptional control mechanism. So in bacteria there are several mechanisms for gene regulation at the level of transcription. That means most of the gene regulation mechanisms are controlled at the level of transcription. And in that there are two systems. First is the positively regulated system and second is negatively regulated system. So in negative regulated system, inhibitor is present. A molecule called as inhibitor is present in a cell and it prevents further transcription. So this is called as negative regulated system. Whereas in positively regulated system, an effector molecule, for example some kind of protein, it activates a promoter which results into high level of transcription. So called as positive regulated system. So let's see with the diagrammatically. This is the positive control in which a protein such as activator which binds to this DNA and it results into the transcription increase. Whereas in the negative control, a protein such as repressor, it will bind to this DNA and results into decrease of transcription. So in the positive control, there is increase in transcription, and in negative control, there is a decrease in transcription. So this is the difference. Let's study the structure of particularly lac operon. So lac operon is present in the E. coli and it contains three structural genes such as lac-Z, lac-Y and lac-A. Lac-Z controls synthesis of beta-galactosidase. Lac-Y controls synthesis of permease and lac-A controls synthesis of trans-acetylase. In addition of that, this operon also contains promoter, operator and cap binding site. Promoter region provides binding site for RNA polymerase. Operator region is negative regulatory site and cap binding site is positive regulatory site. Okay. So this is the diagram of this LAC operon, which consists of these three structural genes, LAC-Z, LAC-Y and LAC-A. Along with that, it also contains this operator and this promoter and this cap site. So whenever there is a repressor is present, it binds to the operator. At that time, it blocks the transcription process. And whenever there is a cap site, okay, so it promotes the RNA polymerase binding. So it will result into the high level of transcription. So in this way, the LAC operon operates. Let's see the mechanism of this LAC operon. So LAC operon expressed only when lactose is present, okay, and glucose is absent. So these are two sugars, lactose and glucose. So LAC operon expressed only when lactose is present and glucose is absent. So there are two regulators which turn the operon on and off condition. Okay, there are two regulators and this regulator will affect in response to lactose and glucose level. These two regulators are LAC repressor and Catabolite activator protein, which is denoted as CAP. Okay, so the LAC repressor acts as a lactose center, and this LAC repressor normally blocks the transcription process, whereas the Catabolite activator protein, that is CAP, it acts as a glucose sensor and generally it activates transcription of operon. First, we will discuss about LAC repressor. So LAC repressor is a protein which represses transcription of LAC operon. So when lactose is not available, LAC repressor binds to operator and it prevents the transcription process. Okay, whereas when lactose is present, so LAC repressor loses its ability to bind the DNA, and so RNA polymerase will slide along the DNA and there will be high level of transcription. Okay, so when LAC repressor loses its ability to bind to DNA, so at that time the change in the LAC repressor, it is caused by a small molecule called as allolactose. Actually, allolactose, it is an isomer of lactose, and this allolactose acts as an inducer which triggers the expression of operon. Okay, let's see diagrammatically. So whenever there is a lactose absent in the medium, so the LAC repressor, it's bind to the operator. So at that time it prevents the transcription process. Whereas when there is a presence of lactose, at that time the repressor will be released from this operator region and there is a allolactose which binds to repressor and released from operator. Now RNA polymerase will transcribe the operon. Okay, so these are two conditions. The second type of regulator, it is the Catabolite activator protein, which is denoted as CAP. So as we know that when lactose is present, RNA polymerase bind to promoter and will transcribe the operon. Okay, but keep in mind that RNA polymerase alone does not bind to lac operon molecule. There is another molecule, that is catabolite activator protein. So, this protein binds to DNA just before the promoter, and it helps the RNA polymerase to attach to promoter, and it will result into high level of transcription. Okay, so this is a positive regulation, but this catabolite activator protein is regulated by a small molecule called as cyclic AMP. So whenever cyclic AMP is there, so it acts as a hunger signal, okay, made by bacteria when glucose levels are low. So at that time, cyclic AMP will bind to CAP, that is catabolite activator protein. It forms a complex and it will be able to bind to DNA, and it promotes the transcription process. Okay, let's see diagrammatically. So there are two situations when glucose levels are low and when glucose levels are high. So when glucose levels are low, cyclic AMP is produced, and this cyclic AMP attaches to catabolite activator protein. They form a complex and allowing it to bind to this DNA. So this cap helps the polymerase to bind to promoter, and it will result into high level of transcription. Whereas in the second case, when there is a high glucose, at that time no cyclic AMP is made. So there is no cyclic AMP and cap complex. So transcription occurs at very low level. Okay, so when glucose levels are low, it results into the high level of transcription, and when glucose levels are high, it will result into low level of transcription by the regulation of catabolite activator protein. So these are the two regulators, lac repressor and catabolite activator protein. But the question is, when does this lac operon turn on? So there are two conditions: when the glucose is not there and when lactose is there. So when glucose is unavailable, at that time cyclic AMP binds to cap region, they form a complex, and it will result into the high level of transcription. Whereas when lactose is there, lac repressor will be released from operator. It will result into the RNA polymerase slides over DNA, and it will also result into the transcription. So in these two cases, when the glucose is unavailable and when the lactose is available, the lac operon is in on condition. Now let's put all the cases together. Let's see how the transcription is regulated. So in the first case, when the glucose is present and lactose is absent. So when lactose is absent, the repressor bind to operator and the RNA polymerase will not slide over the DNA, and so there will be no transcription. Okay. So in the second case, when the glucose is present and also lactose is present. So in that case, the repressor released from operator with the allolactose, and it will result into low level of transcription. Okay, this is the second case. So in the third case, when glucose is absent and lactose also absent, at that time repressor bind to the operator and there will be no transcription. And in the last case, when the glucose is absent but lactose is present, at that time repressor will be released from operator and there will be activation of cyclic AMP and a cap molecule. There is a complex formation and it will result into the strong transcription. Okay, coming to the summary of these lac operon responses. So whenever glucose is there and repressor binds to the operon, it will result into the no transcription. So when glucose is there and lactose is also there, it will result into low level of transcription. When there is binding of cap molecule and repressor, at that time also it will result into no transcription. And finally, when there is a lactose is present and also cap molecule binds to the operon, it will result into strong transcription. Okay, so this is the summary of lac operon responses. So friends, in today's session we have seen the gene regulation in prokaryotes with the example of lac operon. So these are certain references for this topic. So friends, we will stop here and we'll meet in the next session with the gene regulation in eukaryotes. So thank you very much.